What do you mean about mutation? Sudden change in the nucleotide sequence. Sudden change in the nucleotide sequence is called mutation. What do you mean about sudden change? Where it is happening, the sudden change at the nucleotide. What do you mean about nucleotide? Nucleotide consists of nitrogen base. So we need to understand basically nucleotide is a molecule, okay, which is having the nitrogen bases. We know very well the nitrogen base is a structure which is the part of the nucleotide. Okay, nitrogen base plus sugar plus phosphoric acid. So plus phosphoric acid, all this important molecule are the part of the nucleotide. Are the part of the nucleotide. So phosphoric acid, nitrogen base, sugar, all these things are the ingredients of the nucleotide. And we know very well this nucleotide, if it is getting interruption by mutagenic agent, mutagenic mutagenic agent causes what exactly? Mutation. So what do you mean about mutagenic agent? The agent which causes mutation are called mutagenic agent. This mutagenic agent causes mutation. Okay. Uh, we know very well the mutation is caused by mutagenic agent. Okay. Those nucleotide of the DNA which is highly vulnerable for getting mutation is called muton. It's called muton. So muton is a segment of the DNA which is highly vulnerable or highly susceptible or highly prone for getting mutated. Those particular sequence of the DNA is called muton. Okay, muton is nothing but a DNA sequence which is highly vulnerable for getting mutated. Okay, those DNA sequence which is highly vulnerable for getting mutation is called what? Muton. Again, back I'm repeating, DNA which is highly vulnerable or highly susceptible or highly prone for getting mutated is called a muton. Okay, those DNA sequence is highly prone for getting mutation is called muton. Right. So the prone, the word denotes not for all the DNA segment, only for certain DNA segments. Only for certain DNA segments which is highly valuable. Okay, those DNA sequence undergoing mutation are called muton. This muton is not a mutagenic agent. Okay, but this muton is a expressing area in response to the mutagenic agent. Okay, the mutagenic agent are many number. So we know very well the mutagenic agents which are many number, like this we are saying for you, right? So mutagenic agent are many in number, like this we told you, right? So the mutagenic agent or a factor, can you give some example for the mutagenic factors? Means physical factors. Very good example is X-ray, gamma ray, UV ray. Okay, physical factor. Okay, physical factor, chemical factor. Again, back I'm repeating: physical factor, chemical factor, biological factors. Like this, many factors are getting involved in the mutagenic process. Okay, we all need to understand this. The physical agent, chemical agent, or physics agent, biological agent. Can you give an example for this? X-ray, gamma ray, UV ray. So X-ray, gamma ray, UV ray. Okay, ionizing radiation, ionizing radiation. So ionizing radiation, X-ray, gamma ray, UV ray. So these many number of agents are the physical agents. Chemical agents are nothing but nitrous oxide. So mustard gas. If you're talking about the mustard gas, if you're talking about the X-ray, UV ray, so these are all the physical agents. So these are all the chemical agents. Biological agents are nothing but adenovirus. Adenovirus. So adenovirus, baculovirus, sarcomavirus, like this, so many examples we can give for the biological agent. So again, back I'm repeating, adenovirus, okay. So baculovirus, sarcoma virus, okay, sarcoma virus. So sarcoma virus. So what we need to understand is all these virus causes mutation and the potential to cause a mutation, right? So these many number of examples we told for you. So again, back I'm repeating. So in the case of mutation causing agents, physical agents, chemical agents, and biological agents are have the potential to cause a mutation. Not all chemical agents, not all physical agent, not all biological agent. Only few certain agents like X-ray, I told you, chemical agents we already discussed. Okay, even in the case of smoke also, we can say the chemical name called N-nitrosamine, which is very, very important for causing the issues. So we are coming for biological agent, physical agent. Okay, again I'm repeating: physical agent, chemical agent, biological agent. So all these agents causes mutation. All these agents causes mutation. Uh, the These agents are called mutagen. Okay, mutagen. So mutagen. So mutagen. It changes the methyl group or it changes the ethyl group of the DNA. Okay, mutagen. So these all things are okay. Now we are entering into the next important concept, mutation, right? Uh, in the mutation, basically, is caused by mutagenic agent, right? So first thing is point mutation. First thing is point mutation. So what do you mean about point mutation? Any one of you can say this message. What do you mean about point mutation? See, if you're talking about in a DNA, A, T, G, C, C, T, A is it, right? So these are all the nucleotides. These are all the nucleotides. If the nucleotide is getting changed, for example, if the C is becoming G, or if the A is deleted, or if the C is introduced between the T and A, okay, so like this, if the one nucleotide is becoming some other nucleotide, okay, or one nucleotide is deleted or replaced or added, then we are calling that as a mutation, right? So now if you're talking about the mutation causing agent are called mutagen. Mutation causing agent are called mutagen. So there is a point mutation. First is substitutional mutation, another is insertional mutation, another is deletion mutation. One nitrogen base is replaced by the another nitrogen base, one or more base are added and one are removed, okay, but they're not told about additional mutation. So that probability is also there. In a DNA, so many nucleotides are existing. In this so many nucleotides, you can put in any way, in any sequence as you like, but it should be a multiple of A, multiple of T, multiple of G, and multiple of C. You can put in any way according to your wish. And this nucleotide sequence, if it is added with any one nucleotide in between, if any one nucleotide is added in between, then what will happen? That is also called as point mutation. This point mutation denotes this mutation is exclusively for a specific point. If the mutation is happening at a specific point, if the mutation is happening at a specific point, then that is called what? Point mutation. That mutation denotes if one nucleotide is getting deleted, or if it is substituted with other nucleotide, or if it is deleted, substituted, replaced. Okay, it's nothing but substitution and addition. So one more mutation, point mutation, we added as point mutation, right? So now again back in the point mutation itself, you're having furthermore classification information. See, calmly listen to me. Transition point mutation. So you know very well, transition point mutation. So transversion point mutation, transversion point mutation. So what do you mean about transversion point mutation? What is the transition point mutation? In the transition point mutation, if purine is replaced by purine, in the transition point mutation, in the transition point mutation, if purine is replaced with the purine or pyrimidine is replaced with the pyrimidine, then there is called what? Transition point mutation. If purine is replaced with one purine, if pyrimidine is replaced with another pyrimidine, then that is called transition point mutation, right? Okay. So we know very well the purine are nothing but adenine and guanine, right? So pyrimidine are nothing but cytosine and thymine, right? So these all things we know very well. Adenine, okay, guanine, adenine, guanine, okay, cytosine, okay, thymine. So these are all the different type of nucleotides, okay, like this we are not saying these are all the nitrogen bases. So we should be very, very careful when you are writing the information in your answer script, right? So the A and G are purine, okay? These A and G are purine, and C and T are, okay, pyrimidine. So if the purine is replaced by some other purine, if pyrimidine is replaced by some other pyrimidine, then it is called transition point mutation. That is exclusively for one particular point mutation. Now we are coming for transversion point mutation. When I'm going for transversion point mutation, okay, transversion point mutation. So in the transition point mutation, we know very well the purine if it is replaced by pyrimidine. If purine is replaced by pyrimidine, if pyrimidine is replaced by purine, again back I'm repeating, if purine is replaced by pyrimidine, if pyrimidine is replaced by purine, then that is called what? Transversion point mutation. Transversion point mutation. Okay, transversion point mutation. So transition point mutation, transition and transversion point mutation saying exclusively for a specific nucleotide. There are some point mutation we can say for chromosome also instead of saying for nucleotide of the DNA. So what do you mean about that? Okay, here I am saying about translocation point mutation. Translocation point mutation indicates the issue in the one particular segment of the chromosome. So one segment of the chromosome is getting what issue? Again, back I'm repeating, one segment of chromosome is getting the issue. One set of chromosome is getting the issue. Then there is called what? Translocation point mutation, right? So if the chromosome is going for unusual crossing over instead of normal crossing over, for example, unusual crossing over. Can you see this place here? So unusual crossing over. So the unusual crossing over the chromosome, okay, at one particular point of the chromosome indicates translocation point mutation. We can say improper crossing over. Improper crossing over. Again, back I'm repeating. Improper crossing over denotes what? Translocation point mutation. Right. So improper crossing over denotes translocation point mutation. In this particular translocation point mutation, what happens is one segment of the chromosome is exchanging its segment misappropriately with the other chromosomal segment wrongly. Can you see these very lengthy chromosomes, right? So these chromosome sets are going for wrong replacement and exchange of the chromosomal segment. Okay. Translocation in botany, that too in the transport segment, it is having totally different meaning. Translocation denotes the movement of the molecule. Okay, molecule movement. So molecule movement is also called as, again back I'm repeating, molecule movement is also called as translocation. Molecule movement is also called as translocation. Again back I'm repeating, molecule movement is also called as translocation. In the case of plant botany segment, the transport of the starch is called translocation. The transport of the water with mineral, translocation. But here the translocation in the mutation category, totally explaining the different concept representing the misappropriate chromosomal segment exchange. Misappropriate chromosomal segment exchange or unusual chromosomal exchange between the two different length of chromosome, between the two different type of chromosome. This is an unusual crossing over, unusual chromosomal segment exchange. Like this, we need to narrate in the answer script, right? Now, I'm erasing the whiteboard. Now we are coming for the frame shift mutation. Okay, be very careful with this. In the frame shift mutation, if the nucleotide is inserted or deleted, okay, so what will happen is, for example, um, instead of uttering the word, okay, here genetics. Okay, instead of uttering the word genetics, I'm saying genetics. The meaning changes. Instead of uttering the word mutation, mutation, okay, mutation. So the meaning totally changes, right? The way how you are pronouncing, the way how you are uttering the word in a particular context there, meaning exactly totally gives a different value, right? So here, in the same way, for example, when we are giving the technical explanation, when you're using multiple technical vocabulary in any particular event, we are uttering the word. We are repeating the word 2 3 times so that what the concerned receiver will get the appropriate information, right? So either we told, for example, in the case of mutation itself, you just observe this information. I am writing so many technical terms. Um, when the concerned technical executive or the expert while narrating this information, okay, either is saying mutagen, mutagen, or muton, or meton, muton, or mutagen, okay, or mutagenic agent. So he's saying mutagenic agent, or he's saying muton, or he's saying meton, or he's saying mutagen. What is saying exactly? Okay, what is saying exactly? So whenever we are saying multiple terms, mutagen causes mutation. The mutagen and mutagenic agent both are same. Meton, the word we are using in the case of the chromosomal karyographic symbols, but we should not supposed to use in mutation. The muton denotes those DNA sequence which is highly vulnerable or susceptible for getting mutation, right? So we should be very very careful what we are saying exactly, right? So in the case of frame shift, okay, why you told this information when we discuss frame shift mutation? In a frame shift mutation, A, T, G, C, T, A, T is there, but some nucleotide is added here, some nucleotide is deleted here. So what is exactly happening here is AAT is there and G is deleted, CTAT is there. What happens is, what happens exactly? Please listen very, very carefully. For example, AUG, okay, and then GUC. This AUG codes for methionine, GUC codes for valine. So if there is a addition of new nucleotide, if there is a deletion of other nucleotide, what happens? The DNA produces a wrong protein with a wrong amino acid sequence. Why the wrong protein is formed, which is having the malfunction? Why? Because that particular protein is not having a proper amino acid. Why there is no proper amino acid? Why? Because that particular amino acid making RNA is defective. Why the RNA is defective? Why? Because DNA is defective. Why the DNA is defective? Why? Because mutagen causes issue. Mutagen causes issue in the DNA. So DNA causes issue in the RNA. RNA causes issue in the protein. Okay, because of the change in the amino acid sequence. So ultimately, the particular character is not getting expressed appropriately, right? So this is the way you need to understand them. So if we are uttering the word misappropriately, if you are not uttering the word properly, how the meaning changes drastically. In the same way, if there is a change in the nucleotide sequence, what happens is the DNA will make the wrong RNA and the RNA will make the wrong protein. Why there is a wrong protein? Because there is a wrong RNA. Why there is a wrong protein? Why? Because there is a wrong RNA. Why? Because there is a wrong RNA. Why? Because there is a wrong DNA. Right? Wrong DNA, wrong protein. So wrong protein because of the wrong RNA because of the wrong DNA. Right? So this is because of the mutation. This is because of the mutation, right? So uh, here, this protein is responsible for making the enzymes. This protein is responsible for making the hormones. This protein is responsible for making the skin. This protein is responsible for making the hair. This protein is responsible for making the cell membrane. So many issues are arising because of the change in the protein, change in the RNA, change in the DNA because of the change in the mutation impact, right? Change in the mutation impact. When we all studied the social science concept in the lower grade, we know very well in Hiroshima and Nagasaki of Japan, even many of the students of the Hiroshima and Nagasaki and many other multiple uh, population size of various group are experiencing the health issue, experiencing health issue. Why? Because you know very well the US dropped the atomic bomb over Japan, right? Hiroshima and Nagasaki attack, we all know very well. In Gujarat, we know very well, not in Gujarat, sorry, in Bhopal of our MP, Madhya Pradesh, we saw the pesticide industry blast that created great genetic uh, changes in the people who live there. Because of that, many people are coming to the world with genetic disorders. Some people are having the short hand, some people are having the leg, some people are having the short skull, some people are having the short eye. So all these things because of the mutation, right? With the mutation, we can bring the positive thing also. We can bring the negative thing also, right? So what type of negative thing we discussed already? For that, I already had given the example, Bhopal tragedy. I given an example, and then I given the Japan atomic bomb blast that we given an example. What is a positive impact? The positive impact, very good example of the positive impact is, or the mutation is, we know very well our MS Swaminathan developed the salt resistant rice variety. So the salt resistant rice variety is grown in the seashore area to satisfy the growing population size at a very cheap price. For that, the mutation they already done. So there is a positive impact. So positive impact is also there, negative impact is also there. So you know very well the positive impact, you know very well about the negative impact. The positive impact is the rise of various size and quality are developed to satisfy the growing population size. Whereas the negative impact, I already had given the example, Bhopal, strategy, okay, Japan, atomic bomb blast. In addition to this, the bio weapon, okay, in this they're using microorganism to change the gene sequence, right? So that bioweapon, bio war is also a very good example of the mutation negative impact, right? So definitely they will ask in the exam, give some example for the positive mutation impact and negative mutation impact. So you should be in a position to explain everything properly. So the frame shift mutation, why we are saying why? Because if the DNA frame is disturbed, the RNA frame will be disturbed. The RNA frame is disturbed because of the protein frame is disturbed. The protein frame, if it is disturbed, the enzyme frame is disturbed. If the enzyme frame is disturbed, metabolism frame is disturbed. So why there is a metabolic issue? Why? Because there is an enzyme issue. Why there is an enzyme issue? Why? Because there is a protein issue. Why there is a protein issue? Why? Because there is a RNA issue. Why there is a RNA issue? Why? Because there is a DNA issue. Why there is a DNA issue? Why? Because of the nucleotide issue. Why there is a nucleotide issue? Because of the nitrogen base issue. Why there is a nitrogen base issue? Because of the mutagenic agent. Because of the mutagenic agent. So nitrogen base issue reflected in the nucleotide, reflected in the DNA, reflected in the RNA, reflected in the protein, reflected in the enzyme, reflected in the metabolism. So that is why it is called frame shift mutation. Frame shift mutation, right? So for example, one of our family member is getting health issue and admitted in the hospital. So all the members of the family are getting so many psychological disturbances like this. In the same way, here also, if one frame is getting disturbed, all the frames are getting disturbed to a greater level. That is the message we need to understand, right? So now we are coming for the next phase explanation is chromosomal mutation. So in the case of chromosomal mutation, also we can bring the point mutation. In the nucleotide also we can bring the point mutation, right? So, and different type of point mutation we discussed, not only. See, in the chromosomal mutation, if the chromosomes are getting exchanged misappropriately, if the chromosomes are getting misappropriately, like this we told you, right? Can you say specifically if the chromosome segments are getting exchanged misappropriately means what exactly? Can you say specifically heterologous chromosome? What do you mean about heterologous chromosome? Having the allele for various character. If the chromosomes are having the allele for the various character, for example, this chromosome is having the allele for the skin character, and the second chromosome is having the allele for the hair character. Okay, if these chromosomes are getting exchanged, if is getting exchanging, okay, is exchanging the alleles, or if it is getting exchanging its segment, okay, then what will happen is the misappropriate chromosomes are formed. Why there is a formation of abnormal chromosome? Why? Because the abnormal chromosome is formed because of the, again back I'm repeating, why there is a formation of the abnormal chromosome. The reason for the formation of the abnormal chromosome is because of the misappropriate exchange of the allele. So what do you mean about misappropriate allele exchange between the dissimilar chromosome? What do you mean about dissimilar chromosome? Heterologous, hetero, okay, heterologous. So we should be very, very careful in the case of heterologous. So heterologous chromosome or dissimilar chromosomes are exchanging the alleles. Because of that, abnormal chromosomes are formed, which is going to bring the drastic changes in the environment because of the changes in the particular living species, right? So we should be very careful with all these things when we are explaining the answer for this chromosomal mutation, the answer script. Deletion. Can you see this? Uh, this particular blue and yellow is missing in the formed area. Can you see this in the inversion? Okay, the blue came to this place and the yellow came to this place. This is inversion. Duplication, this yellow and red get duplicated and become one more here. There is duplication. In the translocation, misappropriate chromosome segment exchange. So misappropriate, again back I'm repeating, misappropriate chromosome segment exchange. So misappropriate chromosome exchange or the wrong chromosomal exchange brings a chromosomal mutation. Brings a chromosomal mutation. We should know this, all these things, right? So translocation, segment of one chromosome shift to the another non-homologous chromosome. Another non-homologous chromosome. Are you getting my point or not? So one chromosome uh, is getting shifted. The two chromosomes are there. Okay, both are having dissimilar size and shape. Okay, and this chromosome, if it is exchanging the allele, then we are calling as chromosomal mutation. Chromosomal mutation. The into denotes crossing over, right? So when the two dissimilar chromosome, in terms of size, in terms of shape, in terms of allele existence, if it is getting go, it is going. If it is going for improper crossing over, we are experiencing chromosomal mutation, right? Chromosomal mutation. This is the way we need to understand. Now we are coming for the next phase explanation in the chromosomal mutation. What we told about the deletion. If the one chromosome segment is getting deleted, okay, duplication, inversion, translocation, everything we told you, right? Can you say the translocation is because of which particular enzyme? So translocase is an enzyme. This translocase is an enzyme. What it does exactly? Misappropriate segment exchange. Translocase denotes what? Chromosomal segment exchange between the dissimilar chromosomes. Okay. This translocase is also called as abnormal recombinase. See, in the CBSE board exam, can you explain the abnormal recombinase impact like this? Whenever they are saying, we need to say it denotes misappropriate translocation. Misappropriate translocation because of the abnormal enzyme translocase brings abnormal recombination. That is the reason why translocase is also called as abnormal recombinase. This abnormal recombinase or translocase brings misappropriate chromosomal segment exchange or misappropriate allele exchange. That is the way we need to narrate, right, in detail for better understanding. Now we are coming for the next phase of explanation. So this translocase or abnormal recombinase, where it works exactly, if they're asking, we need to understand in the prophase 1 or the meiosis 1. In the prophase 1 or the meiosis 1 or the meiosis, so in meiosis, there are totally two subsegments are there. So again, back I'm repeating, in meiosis, meiosis 2 is present. So in meiosis 2, prophase 1 is present. In the prophase 1, there are many phases are there. So in the packaging stage, of the prophase 1, in the packaging stage of the prophase 1, in the packaging stage of the prophase 1 or the meiosis 2 of the meiosis, this abnormal recombinase is working or translocase is working. So in the CBSE board exam, if you're seeing this chapter very carefully, in the starting of the chapter itself, they told about meiosis and mitosis process with the diagram without explanation. But they have a provision to ask this question. Why? Because they given diagrammatically the explanation. So they can ask this question where this recombinase works. Recombinase works in the pactin of the prophase one or the meiosis one or the meiosis. Okay. So pactin and diprotein both we can include. Why? Because in pactin this event starts and ends in the case of diprotein stage, right? So this is the way we need to understand. Okay. So translocase or abnormal recombinase starts in the pactin stage and ends in the diprotein stage, and then these are the substages of the prophase one, right? So in pactin and diprotein stage, or the prophase one, or the meiosis two, or the meiosis, this translocase works and brings misappropriate chromosomal segment exchange. Like this, we need to bring the concept. Then only your answer secures full mark. Okay. Now, if you're talking about the next major segment in the concept is, if you're talking in detail, uh, here you know very well the chromosome mutations. Chromosome mutations, okay, sometimes reduces the length of the chromosome or increases the length of the chromosome. Reduces or increases the length of the chromosome. Okay, chromosome mutation either increases or reduces the length of the chromosome. Okay, as a result, the gene expression is also getting changed. As a result, gene expression is also getting changed. What do you mean about gene expression? We know very well the gene expression denotes the DNA synthesis process, RNA synthesis process, and protein synthesis process together is called what? Gene expression. What do you mean about DNA synthesis? What do you mean about RNA synthesis? What do you mean about protein synthesis? When you're asking me, we used to call this as replication, right? So DNA synthesis is also called as replication. So the RNA synthesis is also called as what? Transcription, and protein synthesis is also called as translation. So translation, again back I'm repeating, translation denotes protein synthesis process. RNA synthesis is nothing but transcription process. DNA synthesis is nothing but replication process. Like this, we narrated for you, right? So if this gene expression is getting disturbed, the gene expression is getting disturbed. So, so then what will happen is DNA synthesis will not happen properly. RNA synthesis will not happen properly. Protein synthesis will not happen properly. Protein synthesis will not happen properly. So protein synthesis is disturbed. Okay, protein DNA synthesis is disturbed. RNA synthesis is disturbed. Protein synthesis is disturbed. Okay, this is called gene expression disturbances. Gene expression disturbances or central dogma disturbances. Gene expression disturbances or central dogma disturbances. Okay, or genetic flow disturbances. So many number of variety they will ask. Be very careful with this. Central dogma, gene expression. Okay, or allele expression. Sometimes they'll say gene expression, sometimes they'll say allele expression. All these things indicates disturbances in the DNA synthesis, disturbances in the RNA synthesis, disturbances in the protein synthesis. Okay, this is because of the chromosome mutation. This is because of the chromosome mutation, right? So be very careful with all these things when we are explaining the concept in detail. We know very well the chromosome segment. Okay, if it is getting disturbed, these many number of issues are arising, right? So here, non-homologous chromosome is not a homologous chromosome. It is a heterologous, heterologous, heterologous. Like this, we told n number of times for you. What do you mean about hetero? Dissimilar chromosome. What do you mean about dissimilar chromosome? In terms of size, in terms of shape, in terms of the allele distribution. So alleles are not same exactly. Okay, size is not same exactly. Shape is not same exactly. So among those chromosomes, the alleles are getting exchanged. If that is the case, then we are calling that as a translocation point mutation, right? So now we are going for the next phase explanation is causes of mutation. Cause of mutation. Spontaneous mutation. What do you mean about spontaneous mutation? It is happening concurrently. Okay, it is happening okay continuously. Okay, in this what happens exactly? Uh, During the DNA repair process, during the DNA repair process and replication process. Okay, so this will be reversed, okay, in the nature, but it can't be reversed if it is because of the mutation causing agent, right? So yes, of course, okay, here we know very well in the DNA, sometimes what will happen is the enzymes of our normal cell also will do the mistake. Enzymes of a normal cell also will do the mistake. We know very well if the nucleotide is added wrongly, okay, it rectifies its mistake by removing the wrong nucleotide and adds the correct nucleotide in a correct place where it is exactly appropriate. So the deletion I'm writing as minus, the addition I'm writing as plus. So what you need to understand is basically the uh, mutation is bringing the various spontaneous changes in the sense in the nature it happens. There is not a problem at all. Okay, there is not a problem at all if it happens because of the mutation or forced chemical action. Then we can't reverse it in most of the cases, right? External factors, I already given the example, n number of example we given. Not only X-ray, not only UV ray, not only ionizing radiation, X-ray, UV ray, ionizing radiation, X-ray, UV ray, ionizing radiation. Like this, we told n number of information for all of you, right? So now, uh, chemical mutagen. Okay, chemical mutagen. We already told nitrous oxide, mustard gas, N dash nitrosamine, N dash nitrosamine. So N dash nitrosamine is a fantastic example for the chemical mutagen, and this also causes mutation, right? So the gamma symbol, we all know this is the gamma symbol, okay. And then second thing is I some virus, not all virus, I already had given the example adenovirus, baculovirus, okay. Like this we give the example, and even HIV virus is also having the credential to cause the mutation. So one is adeno, another is baculo, another is sarcoma. So these all things whatever I'm writing over the board, please write it down in a note, okay. Why? Because we need to give the elaborate explanation if they're asking a 5 mark, right? Now, we all need to understand basically the mutation causing agent is called mutagen. That mutagen we can use in the lab to get the desirable variety. For example, you know very well our International Agriculture Research Institute, Indian Agriculture Research Institute developed so many number of rice variety of semi-dwarf quality to get developed very faster and to sustain very nicely in the different adverse climatic condition. And also it can tolerate the salt and the water resistant in the sense if it is very salty water also it can grow, okay. Even if the water is having a lot of contamination also it will grow. Even if the water is less also, with that it will go for sustainable growth. All these things because of the change what they already had done in the rice gene by our MS Swaminathan, right? MS Swaminathan alone is not a contributor. N number of agricultural scientists of India developed not only the rice, wheat, maize, like there's so many varieties of grains they developed them by using the controlled mutation. Why? Because the mutation causes the issues in the people who are working in the lab. If you're not following the precautionary measure, the reason behind that is the radiations arising from the particular mutation causing agent causes issues in the body, right? So that is the reason why we should be very careful with all this activity, right? So now we are coming for the next major concept. We know very well the virus is having the causative issue. Causative issue means all virus like this we should not say, only certain virus I told you, including retrovirus. Very good example for the retrovirus is HIV virus. This retrovirus, very good example is HIV virus, this HIV, a retrovirus, and this retrovirus, a type of virus, okay, causes mutation, but all the virus can't cause mutation. All the virus can't cause mutation. This retrovirus causes mutation where? In RNA. In RNA. That means we should understand the mutation can happen in the DNA also, can happen in the RNA also. That is the message we need to understand. Now we are coming for the next major concept is, you know very well, my nose shape controlling genes are there in the DNA. My skull size controlling gene are there in the DNA. So if that is the case, I am calling DNA as a genome, even though I am making RNA. Even though I make RNA, my DNA is called genome. Why? Because my character controlling alleles are there in the DNA. So very good example is human being, right? Okay, now if you're talking about the RNA, okay, the character controlling genes are there in the RNA. If that is the case, we can say the retrovirus is a fantastic example for this. And this retrovirus is having the allele for the character in the RNA. That is the RNA genome. RNA genome, the word I should use only for retrovirus, even though it is making DNA. DNA genome, the word I can use only for the human being, even though it makes RNA. Why? Because in human, character controlling genes are there in the DNA, but not in the RNA. In retrovirus, character controlling genes are there in the RNA, but not in the DNA. This is a message we need to understand, right? So, in the CBSE board exam, they will ask you to give the example for the mutation causing agent for the DNA genome and mutation causing agent for the RNA genome. Mutation causing agent for the DNA genome is human being. Mutation causing agent for the RNA genome is retrovirus. This is a message we need to understand. So twisted questions, tough questions will come repeatedly, okay, in your board paper, right? So what do you mean about genome? Many students are not knowing what is genome. Is different from germplasm. Is different from is different from is different from what exactly? Genome is different. Germplasm is different. Then gene copies are different. Gene copies are different. Gene is different. Gene copies are different. Gene is different. Okay, germplasm is different. Genome is different, right? So germplasm denotes the entire gene content of the seed. The entire gene content of the seed is called germplasm. The entire gene content of your species is called genome. It's called genome. The gene copies are also called as what? Allele. The gene copies are also called as allele. Okay, gene copies are also called as allele. Gene copies are also called as allele, right? Genome denotes the entire gene content of the species. Germplasm denotes the entire gene content of the seed. Gene copies is also called as allele, and gene is totally different. Gene is totally different, right? So here the mutation, okay, mutation they can use for genome, mutation they can use for germplasm, mutation they can use for gene copies, mutation they can use for gene, mutation they can use for DNA genome, mutation they can use for RNA genome. Be competent enough to understand and say the answer correctly for the correct question, right? Now, we know very well the gene mutation is because of the causative agent like physical, chemical, biological, like this. We given the example for you, right? So now, if you are going to the next concept of the mutation, the consequences of the mutation. You know, even now also in Japan, many uh, people are coming with flat nose, pigeon chest, okay, with the irregular skull, okay, and then having the defectiveness in the hand and the leg. Not only in Japan, not only in the um, Bhopal of the Madhya Pradesh, even in other few countries which already undergone a lot of bioweapon impact. So you know very well the plague causing a virus, Yersinia pestis, was spread across many countries during the Second World War. We know very well about that, right? So biological weapon, okay, causing biological weapon ingredients are biological agents. Mutations have no significant impact on organism fitness, okay, and phenotype, and may accumulate in the population over time, accumulate over a population time, right? So sometimes the mutation is visible in first generation. Sometimes the mutation will be not visible for many generation. Okay, it appears after many generation. So this mutation is also influenced by the environmental factors. Very good example of the environmental factors. We told a number of time the climatic condition. For example, we know very well in the China, in Japan, in Philippines, the people are there with less melanin content and having whitish skin because of the less amount of melanin in the skin, right? Okay, whereas if you're talking about Africans, Negroes, okay, we are finding less amount of melanin. No, maximum amount of melanin in African skin. They are having this dark skin because of the huge melanin. Like this, we told this is because of the natural mutation. That is not an issue at all. Okay, that is not an issue at all. Whereas the issue is if you are having albinism. If you are having albinism, albinism is an issue. Okay, if the albumin level in the body, if it is very, very less, we used to call it as albinism. Like this, you don't think melanin pigment is less. If the melanin pigment is less, we are calling as albinism, right? This is a fantastic example of the mutation, and we need to understand basically the genetic disorders or reducing organism fitness. Right? These are all the harmful effect. Now, if you're coming for the significance of the mutation, as I already told to you, developing the grains of high quality with less price by using a defined set of genetic engineering methodology with the involvement of mutation in a controlled manner. If the mutation is executed with a controlled manner, in the sense with the sterile condition, we can bring the effective mutation. That is the reason why we are finding so many varieties of rice, so many varieties of dates, there's so many varieties of camel in the different geographical area of the GCC, right? So we know very well the genetic diversity in the population, which is a driving force in the process of natural selection, right? Development of genetic diversity within the population. Okay. Um, the genetic diversity, of course, it is very, very important if you are getting the superior breed. Okay, what do you mean about superior breed? Some camel can sustain and thrive more nicely in the very tough climatic condition. Some camel can't sustain very nicely in the tough climatic condition. So, sustaining efficiency, which camel is having, that is a superior breed. Which camel is not having sustaining efficiency, there is a defective breed, right? So, this is because of the controlled mutation, right? In addition to all these things, what we need to understand is we know very well the dragon fruits, okay, pomegranate of various size and shape. Um, we are observing among all the apples of the country, various countries. Okay, the Iran apple price is very, very less, but because they are using mutation, okay, and then they are bringing the apple at a very cheap price. Okay, so apple production, rice production, vegetable productions, everything are executed with the gene manipulation by implementing the mutation in a careful environment, right? So we already given the example: dragon fruit, apple, IR at 8, IR at 9, IR at 12, like this. Many varieties of okay, Indian breed varieties are there. Sonolika, Ratan Sona, Jaya, Kalyanika, like this. Many varieties of rice, wheat, maize we are observing, which are all have the potential to grow in a less water area also with a good growth. For that to happen, we need to have a controlled mutation plus appropriate monitoring in the correct interval is very important to get the better variety, right? Now we are coming for the next point is pedigree analysis. Before going to the pedigree analysis, okay, I'll be discussing some few questions of this. Can you see these questions here? Here, the term mutation and sequence of the contest or the genetic evaluation, not evaluation, evolution. Can you see this, please here? Uh, so the way how you are presenting should be like this. Again, back I'm repeating. Explain the term mutation and its significance in the context of genetic and evolution. Okay. For this, you can see the mutation is a permanent and heritable alteration in the DNA sequence of an organism, right? Okay, and significance in genetics that introduce genetic diversity within the population. This is the way the answer should be described in detail, right? So, For 2 mark, we need to explain minimum 3 sentences. We need to give 3 sentences with the appropriate example. Describe the type of point mutation and provide examples and explain how they can impact protein synthesis. So we discussed so many MCQs. Okay, MCQs we are discussing, case studies we are discussing, assessment reasoning we are discussing, previous board question papers we are discussing, structured questions also we are discussing now for having a multiple variety of explanation. Can you see this place here in the explanation? In the point mutation, all these varieties of mutations we need to include substitution, insertion, deletion, etc. Now only we discussed this concept before 10 minutes back, right? So point by point, we need to explain an important word we need to underline. Okay, important word you need to underline. Okay, for better clarity. Okay, with a pencil, so that the consent paper evaluator will feel more comfortable while evaluating your paper. So this paper secures maximum marks, right? So now we are coming for the next important question in the mutation concept. Okay, explain the concept of the frame shift mutation. Provide an example from different point mutation. Can you see this place here? Okay, in this place, frame shift mutation involves insertion or deletion of the bases, altering the reading frame of the genetic code. For example, insertion capital T between A and G in the ATG leads to TATG, right? So like this, we need to give the example. Now only I told now the same examples when I written over the whiteboard. We told this many number of consequences and appropriately the impact. Also, we explained to you in detail with the example, right? That we need to narrate here. Just only we discussed this question in form of content, right? So now we are going for the question number 4 in the mutation. Discuss the role of mutation in evolution. How do they contribute to genetic diversity and adaptation? If you're talking about this, mutation involves genetic variation within the population, which is a raw material for natural selection and evolution, right? Which is raw material for natural selection and evolution. They provide genetic diversity, necessity for the organism to adapt to change the environment, right? So totally, I given totally 3 points for 2 mark, and the 2 a lengthy sentence. Okay, then only you'll get the full marks, right? So be very careful while explaining everything. So now I'm coming for the related videos. Whenever you're having time, please examine in our Go to University portal and then have a better idea. Now point mutation, the way this is the way we need to explain with a lot of segregation, right? So a DNA sequence originally coded for amino acid tyrosine. Due to a mutation, the sequence now read TAT. Explain the type of mutation that occurred and its effects on the protein, right? The mutation in the DNA sequence changes TAC to TAT, a type of point mutation known as substitutional mutation. So like this, what we are explaining the crux and then pinpointing exactly it is a what type of specific frame shift mutation, missense mutation, right? This frame shift came because of missense, right? So the amino acid instead of what? So instead of tyrosine, okay, it is getting histidine amino acid. Instead of tyrosine amino acid, it is getting histidine amino acid, right? So you should be specific in narrating all the examples. This is the way your answer script is supposed to be in the point mutation. And now when we are saying for the frame shift mutation, this is the way your explanation should supposed to be, right? Okay. The DNA sequence, the base pair sequence of CGTCAA was altered to this, identified the type of mutation. Of course, it is a point mutation, okay, or frame shift mutation, but because the protein frame is getting changed, okay. Okay, can you see this? A completely different protein is formed. Okay, there is a very good example of this particular frame shift mutation question. This is the way your answer should be explained while narrating in the board exam. Now, chromosomal mutation. When they are asking, explain the type of mutation involved and discuss the potential impact of the individual. Okay, this is basically a case study question. Can you see this? In a cell karyotype analysis, it was observed that the portion of the chromosome 4 and being attached and reattached to the chromosome 7. This is a case study question. So for this, they're asking the subjective question, and we need to narrate. Okay, chromosomal mutation called translocation. Like this, we need to explain. And then if it is 2 mark, this much is enough. If it is 3 mark, you go from this way, this much sentence explanation to this much. If it is 5 mark, okay, then you go for maximum explanation like this, right?